Welcome, curious minds, to the deep dive. Today we're embarking on a journey into a domain that feels like it's been plucked straight from, some pretty ambitious science fiction.
Hey.
Yet it's unfolding right now, or, well, in our very near future. We're going to plunge into this really fascinating intersection of artificial intelligence and fundamental biology, exploring a place where these cutting-edge digital circuits meet the incredibly intricate dynamic dance of life happening within our own cells.
It's an amazing connection.
Yeah, and that's why we're calling this deep dive Circuits and Society.
That's a really apt title, actually, because our mission today is to explore a truly groundbreaking new AI development. This research is pretty much hot off the presses.
Okay.
From a collaborative team, brilliant minds at MIT, Harvard, and the Broad Institute. And look, we're not talking about just an incremental step forward here. No, this feels like a quantum leap. It's poised to fundamentally revolutionize how we understand, diagnose, and ultimately treat a whole vast array of diseases.
How? By changing how we map things.
Exactly, by changing how we map the very building blocks of human life, our proteins.
And what's the core problem they're tackling, you might ask?
Well, it all boils down to proteins, as you said. These vital molecular machines, they're absolutely everywhere in our cells, doing countless critical jobs.
Absolutely essential.
But here's the mind-boggling kicker. A single human cell can contain around 70,000 different proteins and their variants. 70,000.
Staggering number.
Now, imagine trying to figure out where each one of those proteins is located within the cell, manually.
Yeah, forget it.
That's not just a needle in a haystack. It's like trying to pinpoint a specific grain of sand on, every beach in the world. It's incredibly costly. Takes an extraordinary amount of time, and its scope is just severely limited by, human capacity.
And this isn't merely some, academic challenge. It has real consequences. Proteins that end up in the wrong place or are mislocalized, they're directly implicated in some of the most devastating diseases we face.
Like what? Well, we're talking conditions like Alzheimer's, where misfolded proteins accumulate, cystic fibrosis, caused by errors in a specific protein's location and function,
Right.
and various forms of cancer where cellular anarchy often begins with protein dysfunction. So pinpointing their exact granular location is absolutely crucial.
Crucial for understanding how these diseases develop and how to fight them.
Exactly, how they progress, and critically, how we can effectively fight them. The current manual methods, they're just too slow and too expensive to make really rapid breakthroughs.
Okay, so the big tease then, the incredible solution we're diving into today. A new AI model.
Right.
One that completely bypasses these traditional laborious limitations.
A shortcut.
Yeah, it offers a shortcut, a fast lane, to a level of critical biological understanding we've maybe only dreamed of. This research is so cutting edge, so timely. You might even say it's, from the future.
Maybe.
So stick with us, because this deep dive is going to reveal some truly astonishing insights. Okay, let's really unpack the cellular landscape and how AI is now stepping in to map it with, unprecedented precision.
The sheer scale of the challenge has always been a major bottleneck, right, for biological research.
Absolutely, a huge bottleneck. To truly grasp this scale, think about one of the largest existing data sets for protein location, the Human Protein Atlas.
Right, I've heard of that. Massive project.
It is massive, an international effort aiming to map every human protein's location. It's immense, cataloging over 13,000 proteins across more than 40 different human cell lines.
Okay, sounds impressive.
It is. But even with all that incredible data, it has only explored a tiny fraction, a mere 0.25% of all possible protein cell line pairings within its own database.
Wow, 0.25%, that's it?
That's it. And the vast majority of existing AI models, they're constrained by what they've already seen, meaning they can only really generalize within the data they were trained on, or they just average results across different cell types, which loses all the important detail.
Exactly. It completely loses the crucial individual detail and nuance of specific cellular environments.
So it's not just the sheer volume of data, it's the monumental human effort involved too. Like you said, scientists, even with all their dedication and tech, can only realistically test and validate a handful of proteins at a time.
It's painstaking work.
It's like trying to explore the entire ocean by just, dipping your toes in a few specific spots.
Yeah.
Imagine being a cancer researcher, knowing there's a vital protein involved in a patient's specific tumor response, but needing months, maybe even years, just to manually locate it in different cell samples. That's been the frustrating reality.
It has, and this is precisely where AI steps in. It's offering not just a magnifying glass, but more like a wide-angle, ultra-high-resolution lens for researchers.
And the name of this groundbreaking AI is PUPS. Now, for our listeners, PUPS stands for Prediction of Unseen Protein Subcellular Localization.
Right.
Sounds a bit technical, I know, but the implications, as we're about to discover, are anything but complicated. They're huge.
That's a great way to put it, and you're right. This isn't just some incremental tweak to existing methods. Not at all.
So what makes it revolutionary?
What makes PUPS truly revolutionary is its unprecedented ability to predict the location of any protein.
Any protein?
In any human cell line.
Wow.
Even if the model has never encountered that specific protein or that specific cell line before during its training.
Okay, hold on. That's the key part, isn't it? Even if it's never seen either before.
That's the phenomenal leap in generalization and predictive power. It's a game changer.
Wow. Okay. So it's not just recognizing patterns it's been shown. It's truly inferring from, like, first principles somehow. And you mentioned single-cell level localization. Why is that such a big deal compared to just an average estimate? I mean, aren't cells pretty much the same within a specific line?
That's a great question, and it really gets to the heart of why this is so important. See, while cells in a line might appear similar, they're incredibly dynamic.
Okay.
They can respond uniquely to various conditions, think a drug, an infection, or even just stress. Average estimates might tell you where a protein generally is across a whole population of cells.
Right, the average location.
But that average can hide critical variations between individual cells. Single-cell localization gives you those precise, granular insights.
Give me an example, like in a drug trial.
Exactly. Perfect example. Imagine a drug trial. You could pinpoint a protein's exact location in one specific cancer cell after it's been treated.
Okay.
See how the drug affected that individual cell's internal machinery? Instead of just getting a blurry average across thousands of cells.
That makes sense. Much more detail.
Right. That level of detail can unlock understandings of, say, disease progression, treatment effectiveness, even things like drug resistance that were pretty much invisible to us before.
So the million-dollar question then. How does PUPS actually do this? What are the brains behind this breakthrough? How does it make such nuanced predictions for stuff it hasn't seen? It sounds, I don't know, almost like magic.
It's not magic, but it is incredibly clever. PUPS uses this sophisticated two-part method. It combines different types of AI models, each bringing a unique piece of the puzzle.
Okay, two parts. What are they?
First, there's what they call a protein sequence model. You can think of this like a specialized protein language model.
A language model, like for text, but for proteins.
Kind of, yeah. It analyzes the protein's amino acid sequence. That's essentially its genetic blueprint, right? Its unique string of molecular letters.
Got it.
From this sequence alone, the model can understand the protein's inherent localization properties. It can even infer things about its 3D structure.
Wow.
It's like teaching a computer the grammar of proteins, understanding how their precise molecular composition dictates where they prefer to sort of hang out within a cell.
Okay, so it understands the protein's own intrinsic properties, its molecular identity. What's the other half of this power couple?
The second part is an image in-painting model. This is a special type of computer vision AI.
Computer vision. Huh.
This component acts like a cellular detective. It provides the environmental context. It takes 3 different stained images of a cell.
Stained images, like in a lab.
Exactly. Specifically images of the cell's nucleus, its control center,
Right.
its microtubules, part of the cell's internal scaffolding and transport system,
Okay.
and its endoplasmic reticulum, or ER, which is a vital network involved in making proteins and lipids.
So just those 3 images.
Just those 3. From those, this model gathers vital information about the cell's state. It effectively understands the specific cell type, its unique individual features, and even if it's currently under stress from disease or something else.
That's a fascinating duality. So it's looking at the protein's inherent characteristics and the specific unique environment of the cell it's in. And then these 2 completely different pieces of info. The language model, understanding the sequence, the vision model, understanding the cell's state, they somehow come together. How does it actually join those representations?
Exactly. So once both models have processed their respective data, the protein's intrinsic grammar from its sequence, and the cell's unique state from its internal structures, PUPS then integrates these 2 streams of information seamlessly.
It merges them.
Yeah, but it's not just like overlaying them. It's creating a unified predictive map. Think of it like having the detailed blueprint of a specific building component, right? And a
Right.
precise understanding of a particular piece of land, then instantly combining them to find the exact coordinates of where that component would fit best or wouldn't fit on that terrain.
Ah, I see.
This allows it to output a highlighted image, clearly showing the predicted location of that specific protein within that specific single cell. It even accounts for subtle differences between individual cells in the same culture.
That's an incredibly clever approach. But teaching an AI to be such a sophisticated cellular detective, especially to predict the unseen, that sounds like a complex training process. This is where it gets really interesting, I bet.
It is pretty interesting.
What specific tricks or innovations did the researchers use to get PUPS to generalize so effectively to things it had never encountered before?
They employed some truly ingenious strategies during training, really pushed its capabilities far beyond typical models. One key trick was assigning PUPS a secondary task during its training phase.
A secondary task, like homework.
Sort of. In addition to its main goal, predicting the protein's location as an image, PUPS was also explicitly asked to name the specific cellular compartment.
Name it, like nucleus or ER.
Exactly, like the cell nucleus or the endoplasmic reticulum. This forced the model to not just see the location, but to actually understand and categorize it. The paper used an analogy,
Yeah.
like a teacher asking students to not just draw parts of a flower, but also write down their names.
Okay, so it's not just about outputting a visual, but also reinforcing its conceptual understanding of where things are supposed to be. That sounds like it would build a much more robust internal model.
Precisely. That extra step significantly improved the model's general understanding of all the possible cell compartments. It built a deeper, more abstract comprehension of cellular architecture.
Makes sense. What else?
Another crucial aspect was joint training.
Joint training.
Yeah, unlike many models that might be trained on proteins first, then maybe cell lines separately, PUPS was trained on both proteins and cell lines simultaneously, right from the start.
Ah, so together.
Together, this truly deepened its understanding of where proteins tend to localize across a wide variety of cellular images and conditions. It learned the interplay between protein characteristics and cellular context right from the beginning.
So it's not just learning about proteins in isolation, and not just learning about cell types in isolation, but how proteins behave within those different diverse cell types.
Yeah.
All at once. That sounds much more holistic.
It absolutely is. And beyond that, the model can independently understand how different parts of a protein's amino acid sequence contribute separately to its overall localization.
Wow.
But what's truly remarkable about this generalization aspect, unlike most existing methods that need you to already have a stained image of the protein in your training data,
Right.
PUPS can predict for completely unseen proteins.
That's a huge part of it.
It is huge, because it means it can capture changes in localization driven by unique protein mutations, things that might not even be included in current large databases like the Human Protein Atlas.
So like a rare mutation causing a problem.
Exactly. If a rare mutation in a protein causes it to mislocalize in a specific type of tumor, for instance, PUPS could potentially predict that mislocalization, even if that specific mutated protein has never been observed or cataloged before.
That's a massive step forward for understanding what's happening in really specific, often rare disease contexts.
It really is a massive step. So if we connect this to the bigger picture, what does all this mean for us, for medicine, for, the fundamental understanding of life? The impact and applications of PUPS are truly immense.
I can already imagine some possibilities. From just an efficiency standpoint, what's the immediate gain for scientists in the lab today?
Well, Yutong Seo, one of the co-lead authors of this research, put it perfectly. She said this could save yourself months of effort in lab work.
Months.
Yeah. Instead of manually testing potentially hundreds or thousands of possibilities, PPS can act as this incredibly powerful initial screening tool.
So it guides the experiments.
Pretty much.
Yeah.
Researchers can feed in a protein sequence and a cell type, get a strong prediction of where it should be, and then focus their precious experimental resources on just validating the most promising or maybe the most surprising predictions.
That massively accelerates the discovery process for new disease mechanisms or potential drug targets.
Massively. And in terms of direct medical advancements, how do you see this playing out?
Well, disease diagnosis, maybe. Could it make that faster?
It could lead to much more efficient diagnosis, absolutely. Imagine a doctor seeing a suspicious cellular change in a biopsy.
Okay.
PUPS could rapidly screen for mislocalized proteins associated with specific illnesses, providing crucial diagnostic clues much, much faster than current methods.
That's huge. What about treatments?
It opens immense new doors to identifying novel drug targets. Understanding a protein's precise location and function is fundamental, right?
Right.
It's foundational to developing therapies that can maybe put it back where it belongs or block its harmful activity if it's in the wrong place. We could design drugs with much greater precision.
And beyond the immediate medical stuff, what about just basic biology, just understanding how life works?
It provides a much deeper, more nuanced understanding of how complex biological processes are intimately related to protein localization. We're talking about untangling some of the most intricate mysteries of the cell.
Like how cells respond to stress, or how they differentiate.
Exactly how they communicate, all these fundamental processes. And importantly, the researchers rigorously verified PUPS with actual lab experiments.
So it's not just theoretical.
Not at all. They showed that its predictions consistently had significantly less error compared to baseline AI methods. It confirmed its practical accuracy. This isn't just theoretical, it works.
Okay, so it works now. What's next for PUPS?
Yeah.
Where do the researchers see this incredible technology going from here? They must have future plans.
Oh, absolutely. Their future goals for PUPS are nothing short of ambitious. They want it to evolve beyond just individual protein localization.
To what?
To understand complex protein interactions, how different proteins work together or maybe interfere with each other within the cell.
You're seeing the whole network.
Exactly. They also aim to make predictions for multiple proteins simultaneously within a single cell, painting an even more complete dynamic picture of that cellular landscape.
Wow.
And in the longer term, the ultimate, truly transformative goal is to enable PUPS to make predictions of protein localization not just in cultured cells in a dish.
But where?
In living human tissue.
Living tissue, like in a patient sample.
Imagine the diagnostic power of that. In real time, on actual patient samples, that's the moonshot.
Wow, predicting protein locations in living tissue. That truly feels like the future of personalized medicine and biological understanding.
Speaking of the future, something just occurred to me while we were talking about this cutting-edge research.
Sure. What's up?
I was just double-checking the source material, our notes for this deep dive, and the publication date for this incredible paper in Nature Methods. It's listed as May 15th, 2025.
Wait, did you just say May 15th, 2025?
I did. And our current date, as we're recording this and diving into these sources, it's August 8th, 2025.
Fascinating. So, we're discussing a scientific breakthrough that was, from our current perspective, published just, a couple of months ago?
Yeah, like less than 3 months ago.
That explains why it feels so incredibly fresh and forward-thinking. It's literally hot off the digital press. Right. I mean, for a second there I thought, are we time travelers? Or is AI that good at predicting the future of its own research outcomes?
Well, maybe not quite time travel.
No, but this really does make the content even more exciting, doesn't it? It's literally ahead of its time for most people just hearing about it now. We truly are at the forefront, diving into the very latest discoveries as they're happening.
Or, well, as they just happened. It's pretty cool.
Very cool. So here we are at the end of another deep dive, truly marveling at how artificial intelligence is not just changing computing, but quite literally reshaping our understanding of life itself.
Yeah.
It's blurring those lines, isn't it, between circuits and society, or, in this case, between advanced algorithms and fundamental biology, revealing secrets that have been hidden for millennia.
And it raises this important and, frankly, profound question: what other biological mysteries, currently just obscured by sheer complexity and scale, what else could be unlocked by AI that can see and understand at a level that was unimaginable just a few short years ago?
Right. What else is hiding in plain sight, biologically speaking?
Exactly. How will this fundamentally shift our entire approach to health and disease, especially if these predictions become even more immediate, maybe even anticipatory? The potential is truly staggering. It hints at a future where we understand cellular life in ways we've only, dreamed about.
Indeed. If you're as fascinated by these leaps of scientific imagination as we are, make sure you subscribe to The Deep Dive wherever you get your podcasts.
Definitely.
Stay tuned for our next deep dive, and perhaps even the next viral AI conversation, because who knows what future discoveries we'll stumble upon next.